So hi everyone, and first I would like to thank the organization to let me the opportunity to present you today a pitch. So it's a platform, an integrated cellular platform for enhanced preclinical model fidelity in drug discovery that we made at iStem. So first I would like to introduce you a bit iStem. So we are experts in therapy repurposing and also cell therapy, but this platform focuses on the therapy repurposing. Regarding the research areas, we have several research areas such as genodermatosis, neuroplasticity, neuromuscular disease, retinopathies, and muscular dystrophies. And this allows us to have a really large expertise and to to master a lot of large scope, of differentiations for the cells differentiation. And to support our researches and also we made a platform to support our researches, and this allowed us to have 4 packages that we can use on an individual basis or all together. And those packages are disease and LC model creations, mechanistic analysis, therapeutic screening, and lead compound profiling. So I will go through all those packages one by one. So the first one is the biological resource creations, starting usually from the patient cells that we will reprogram to obtain induced pluripotent stem cells. Then we could do genome editing, and then we have the differentiations part, and we will go through quality control, clone selection, and automated bioproduction to obtain our working cell bank. And we will also have other cell banks such as the seed and the master bank, cell bank, and the multiple quality controls and reports for the culture and the genome editing. Once we have our cells, we can do mechanistic analysis. So for this, we will have first the cell imaging with multiparametric analysis and medium to high content screening with the devices such as the IncuSites and the spinning disk. On the other side, we also have the next generation sequencing with so we can do bulk and single cell analysis with NextSeq and equipment controller, for example. And the goal of this package is to verify the phenocopy, to identify the phenotype of the disease, and also to identify the readout for the high throughput screening. And speaking of high throughput screening, this is the next package, so drug screening and hit identification. So it starts with the cell culture miniaturizations. Once we did it, we can do the drug delivery. So we can do several therapies. So ATMPs with gene therapy, viral and non-viral. Also ASO, small molecules with your library or our library. And also recombinant proteins and recombinant antibodies. And for this, the goal is to obtain the lead compounds, to select the lead compounds with this. And we're using acoustic handling device called ECHO that allows us to use only nanoliters to treat the cells and also to do dose response and co-treatment experiments. So once we have the lead compounds, we can do the lead compound profiling. So for that, the main part is usually the mechanism of actions identifications. And so we could do, for example, cell imaging on 2D or 3D models. We could also do pathway determinations with pathway knockout and next generation sequencing to analyze drugs impact. And the goal is really to lead the lead compounds to pharmacological and clinical development. And all those packages can be done on an individual basis or all together, making an end-to-end service to de-risk a project. So now I will show you an example that was made with an iStem. So it was the identification of digoxin as a lead compound from the treatment of DM1 using stem cell-based drug screening. So in that case, we use human embryonic stem cells to make a mesodermal stem cell bank. So that's the first package. Then we did mechanistic analysis, and one of the results was the readout that we will use for the screening. That was the number of nuclear foci and also the size of those foci. And then we were able to screen over 12,000 compounds, and we ended with one compound that's our lead compound, digoxin. Once we had our digoxin as a lead compound, we could do the compound validations. So in these studies, they went through several experiments, starting with microRNA splicing to identify modifications. So there were 44 of them, and 10 were positive ones. Then they tried to understand the mechanism of action of digoxin, And one of the results is that the digoxin mechanism of action is calcium dependent. They also did some studies to determine the best digoxin concentrations. And for that, they use DM1 myoblast fusion. So they studied the DM1 myoblast fusion, and they ended with in vivo studies that were made outside of the platform. So, as you may have understood, it was first developed to support in-house projects. But as we proved the effectiveness within our organizations, we decided to open it to collaborations to enhance our functionality through partnership and also to increase value through shared expertise. And now we are actually developing a fee-for-service approach to offer tailored services from individual basis package to end-to-end services. And I would like to thank everyone who is involved in these projects, so the research teams, the platform manager, the core product team, and the funding entities. Thank you.
Thanks, Aubin. Any questions? Seems everyone is super clear. Yes, yes.
Thank you for your talk. I was wondering, in your mechanistic analysis, when you see or you showed that you used to characterize the phenotype, cell imaging, transcriptomics. So I'm wondering, why don't you push it further, or have you tried to push it further with also proteomics and looking at, because your readout could be the overexpression of a cell, and then you could use that later for your screening.
Indeed. In my presentation, I really showed the basis of the of our platform, but it's really tailored. So depending on the needs of the project, we could also think about pushing further, as you say, the mechanistic analysis. And yeah, it's really a tailored services, so it could be done. But on the basis of the platform, we focusing on those parts I just presented. But thank you for the question.
Thanks, Aubin.